Now I'd like to introduce today's speaker, Dr. Ethan Bier. Dr. Bier is a distinguished professor and holds a UCSD Tata Chancellor's Endowed Professorship in the Cell and Developmental Biology section at the University of California, San Diego. In addition, he's also an Alfred P. Sloan and Basil O'Connor Scholar. He's an Allen Distinguished Investigator, and he's also the Science Director of the Tata Institute for Genetics and Society. Ethan has been studying the genetic and biochemical basis of patterning processes during development and has used the fruit fly as an experimental system. He's also using the fruit fly system to study mechanisms of human disease. Notable in the context of today's webinar, Ethan is also a creative and productive innovator in the gene drive research and development space, where he has developed a number of gene drive systems with unique and useful characteristics. He has been thinking about and developing gene drive control systems, and we're going to hear about that today. So Ethan, welcome. Thanks for joining us today. I know you're busy. At this point, I'm going to turn the floor over to you.
And thanks, David, and the organizers for inviting me to give this webinar. And I'll talk a little bit today about, as David says, some of the lessons we've learned, both about gene drives, actually, and about these elements that we've developed to control them, and about the process, and about some of the, their performance behavior characteristics, and also some questions that we will look forward to trying to address in the future. So we were collaborating with the group of Tony James up at UCI to develop one of the first gene drives. In fact, it was the first gene drive in mosquitoes, that CRISPR-based gene drives in mosquitoes that introduced these anti-malarial cassettes. So this is in the mode of modification of the insects. As you are aware, there are two different strategies for using gene drives to control malaria. One is just to get rid of the mosquitoes, and the Imperial College group has done wonderful work in that domain. And Tony James's group has focused their efforts on developing these sort of sophisticated gene cassettes that get activated upon a blood meal, turn on the expression of anti-malarial effectors, and as a consequence, block the disease. And in the first prototype version that we published, it worked very well through males, but it didn't work so well through females. And the essence of why it doesn't work so well through females, which is an important sort of conceptual feature of the gene drive field, is that in females, the Cas9 and the guide RNA complex that persists from the mother into the egg. And so as soon as the paternal chromosome fertilizes the egg at fertilization, it becomes a target for that Cas9 cleavage. And if that happens before the cells enter the germline, where they're protected, and where at that point you get homology directed repair that does the gene drive conversion that you'd like to see happen. But if it doesn't, if before that you get this mutagenesis taking place, cleavage without copying, then that slows the process. And in fact, it was much less in females than it was in males. And so Valentino Gantz, the person who designed these drives in my lab, came up with a swap construct that has two guide RNAs that are injected together with a construct that has a recoded version of the KH and a GFP gene in here that we can swap for red. So you go from green to red. And when you do, now the gene drive, which in the original construct here disrupted the function of the gene and caused a wide-eyed phenotype and sterility actually in females, was now capable of having a recoded version in there instead. And with the recoded version, you got wild type function. So now the gene drive is protected from making any phenotype, whereas the mutations that get generated by the non-copying events go on persisting to create that phenotype. And in fact, that phenotype turns out to be dominant, because what happens there is if you inherit the mutant allele, the non-drive allele, then that's transmitted by the mother. Then the father's allele again gets attacked, and you get the animal being a facultative homozygote or a mosaic homozygote. We call that lethal mosaicism, or in this case, sterile mosaicism, because the phenotype is sterility, female sterility. But the point is that the gene drive that is recoded is protected from that, and the mutant allele is not. So it's a way of sort of, it's like a catalytic converter in an automobile, gets rid of the pollution or mistakes right away. So just to give you a sense of what this meant in terms of drive experiments, this was the original allele that was not recoded. And in Tony James's lab, they did a test of this, how well it worked in cages. And despite the fact that it didn't drive so well in females and had this fitness cost, it did drive pretty consistently at 1-to-1 ratios into the cage, but then it would either stall or it would go extinct. And in the lower numbers, it would tend to fall out very consistently. And the reason is that there were mutant alleles that got generated that were wild type in function, and then they would outcompete the cassette, because the cassette, as I mentioned, is an interruption of the gene. But the recoded version wouldn't suffer from that. And so Adriana Adolfi in Tony's lab, in collaboration with us that built it, tested this in cage trials as well. And as predicted, the gene drive drove to fairly complete introgression. And you probably know from some of the work from Tony's lab that they also have a gene drive in there in Gamby, Anopheles Gamby. This is in Anopheles Stevens side of these experiments that drives very well in a eye pigmentation gene, a different eye pigmentation gene called cardinal. And there, for reasons that are not entirely clear, but perhaps have to do with the design of the construct that they carefully built, generates almost none of these mutant alleles, these so-called cleavage-resistant alleles. And as a consequence, it drives really well. So the point I'd like to leave with you here is that we've kind of gone from the prototype gene drive concept, showing that it can work in principle, to deriving several different strategies for having the gene drive actually work. And as you probably are aware, there's work done from Andrea Crisanti's lab, where they have similarly developed a very robust gene drive that works on the suppression side, that is to introgress a trait into the population that causes females not to develop properly in that particular case, but again, a negative trait that impacts the ability of the mosquito to either survive or thrive, and that leads to population suppression. And so both of those strategies now have gone sort of from the drawing board initial stage to the stage where there are very effective drives that in principle, subject to the proper negotiations and discussions and inclusion of everybody into the discussion, could be considered for actual field trial deployment. And as part of that, one also wants to develop these target product profiles. And as part of those target product profiles, it'd be very good also to have mitigation strategies at least outlined and perhaps developed, so that one could see, as David pointed out, whether or not there are tools available to limit the spread of these autonomous elements should some need arise to do so. And one of the things that was kind of cool about this particular gene drive is, remember, I mentioned that it works based on this principle of lethal sterile mosaicism, wherein if the parent, the mother, transmits the mutant allele rather than the gene cassette to the offspring, the paternal chromosome gets mutagenized, causing a phenotype which is both white eyes and sterility. And you can see the white eyes, and you can see that at the point where the rate of change is greatest in the drive process, there's a burst of white eyes that is consistent with this generation of these mutant alleles. That's basically the mechanism that's cleaning up. The autocatalytic converter is the catalytic converter is working at that point. There's another strategy wherein you have a gene drive at one locus and you use it to also carry a second guide RNA that biases the inheritance of a second allele, one of which is desired, the one that can't get cut, and the one which is not preferred is the one that can get cut. So the not preferred one could either be a mutant form of the sodium ion channel, the voltage-gated sodium ion channel, the so-called KDR mutants that confer resistance to insecticides, or it could be a parasite susceptible form of an endogenous host protein, such as the FREP1 gene, which comes in two flavors, one that is very friendly to parasites, and that's the typical one, and another one where you mutate a particular residue from leucine to glutamine that confers a refractory parasite phenotype. So you'd prefer the refractory phenotype over the susceptible phenotype for the parasite, and you prefer the susceptible or sensitive phenotype for insecticide resistance for that trait. And so I'll give you an example of one, and we did this in fruit flies, and this is the work of Baghiyashri and colleagues, including several that I'll mention at the end, that have developed this technology. Annabelle Guichard being one of the people who developed the whole concept of it, and Raja Kushwaha, who was a co-first author with Baghiyashri on the paper that we have under consideration on this topic. And what they did first, or what Raja did first, is he made a tagged mutant allele of the gene, the voltage-gated sodium ion channel gene that contained the 1014F mutation, which is the KDR mutation that confers resistance to DDT and pyrethroid insecticides. They showed that it was, in fact, DDT resistant. The 1014F allele was DDT resistant over a wide range of concentrations. We typically use the lower dose for our studies. That it also reduced the lifespan, so it had a fitness cost associated with it, and that it also was very temperature sensitive. And this may actually explain why, although it's a very prevalent mutation, the 1014F mutation occurs, it's been generated, I think, independently about a dozen times in Africa in different locations, in mosquitoes, and it's present in many other insects, but it's never been recovered from the wild in fruit flies. And we think maybe the reason is because it's very temperature sensitive in fruit flies. And so at warm temperatures, the flies would die. And so it's got a very strong fitness cost in flies. And it does carry a fitness cost in other insects too, perhaps partially for this reason. In any case, they generated a drive element, in this case, a split drive element where the Cas9 was provided elsewhere, that has a guide RNA for copying the element and another guide RNA that targets cleavage to the 1014F, but not to the wild type insecticide susceptible 1014L allele. And so they combined that in a starting cage that was 83% DDT resistant and 17% wild type, with 25% of the individuals having the drive element. And over the course of 10 generations, the drive drove to a modest degree of introduction due to the fact that it also generates some of these mutant alleles that limit it going to completion, but it still goes pretty well. And you see that that doesn't happen in the absence of Cas9 controls. And then the question is at different generations, what is the DDT resistance profile? And the DDT resistance profile dropped from 83% to roughly 50 to 40% just by virtue of the fact that there's a fitness cost that's associated with the DDT resistant allele over the wild type allele, but then there's a very significant additional effect of the drive element doing its part. And if you look at the alleles at the end, you see that you've gone down now, you've reversed it from it started off at 83% resistant, now it's only 13% and 87% wild type. And an interesting feature of that is that there's very low variance in this is a sequence analysis, a deep sequence analysis of this trait. Compare it, for example, to the control where there's more typical variants. And we've seen this happen, and I'll mention it perhaps, if I can remember, with the ERACRs experiments that I'll tell you about in a moment, which is that if you have a cooperation of two different drive processes, it results in greatly reduced variance. And the reason is because you have two independent ways of driving. And so there are two independent ways in which the drive process could fail. And it's the failure probability is the probability of failing in both ways. And so that narrows the variation. And so that's an important feature too, because you can really rely on the behavior of a drive that works on two different drive principles. That is, there are two different mechanistic selective advantages given to the drive. Then it really works very well. So then getting to the mitigation part of the talk, these are the two people, Emily Bulger and Shannon Zhu, who were the prime leaders of the project. But as I'll mention at the end, there was quite a big team of people who worked on this. This was, I estimate, about 16 person years of work in the lab. So this was a project we devoted an enormous amount of attention to. And again, largely for reasons that David outlined at the beginning. And so the way this works is you've got guide RNAs, not G2 and G3, but no Cas9. And the eraser is inserted. It's got, this is a backronym for what it obviously does, which is to erase the gene drive, but it's inserted exactly at the same site in the genome as the gene drive. And so what happens then is if you get an eraser carrying insect that mates with a gene drive insect, then what happens is that the Cas9 that's produced by the gene drive combines with the guide RNA produced in trans on this side and similarly on the other side. And now those guide RNAs are programmed to cut on either side of the gene drive, basically deleting it. And then you have the homology directed repair pathway that will copy the material that's in between into the gap. And so the result is you have only erasers that can be transmitted to the progeny, having deleted and replaced the gene drive. And the idea is that if a gene drive, say, went through a population to completion, and then at that point you introduced an eraser should drive with the same kind of kinetics, logistic growth kinetics, as the gene drive does in a naive population if the eraser is introduced into a gene drive population. And the gene drive and the eraser, like the gene drive, could carry anti-malarial effectors. And after sweeping through the gene drive population, the eraser should freeze the process. And so that's the concept that we wanted to test. And so what the collaborating co-authors did on this is they started off with a gene drive that was inserted into the yellow locus. And this is the gene drive element that's got the Cas9 and the guide RNA for copying itself. And then two different erasers, one that, like the gene drive, had a yellow minus phenotype. That is, it interrupted the function of the gene. Just like the insertion we put in the KH locus in mosquitoes, where it interrupted the function of the KH gene, which was required for eye pigmentation and female fertility in that case as well. And so in one case, we had an eraser min, which is just literally like the diagram. It's got two guide RNAs and a marker on it. That's it. And then the other one, there are two versions of this, but the most elaborate version of it was ERACR-2, where we have a recoded version of the yellow gene. So we reestablished function of the yellow. And then we also did everything we could to minimize potential recombination between the eraser and the gene drive, because our preliminary experiment suggested that could be an issue. And it turns out that is true. But ERACR-2 we did everything we could at the time to reduce that target sequence homology. An important point here is that although yellow is just a pigmentation phenotype, it does result in yellow minus males having a great fitness disadvantage with wild type females. The basis for that is not entirely understood, but it's a very strong competitive disadvantage they have in terms of mating. And we verified that in our own experiments that was true as well. It's one of the classic examples from the literature. Actually goes all the way back to Sturdivant, his experiments describing that. But in any case, what we also did is we went through some pains to label all the different components as best we could. So of course, we labeled the eraser in one color and the gene drive in another, but we also associated a tightly linked marker, the white eye color marker, to them. So the eraser was closely linked to the white minus phenotype and the gene drive to orange white plus phenotype. And this allowed us to dissect the process in detail, a very important component of the experimental design. So for example, if we're looking in females, if we're looking at the F2 generation, you can look at the donor chromosome, which will be typically the eraser with its closely linked marker, white minus, and we can look at the target chromosome, the gene drive chromosome, to see what happened to it. So if the eraser did what it's supposed to do and just copied to it, deleted it and took it out at this step, then the result should be the eraser that copied. If it didn't work and it just left the eraser in place, then it should just have the green phenotype. These ones with the orange eyes should just have the green phenotype. If it took it out but didn't copy, it would have no eye color phenotype, but would have the orange eye phenotype. And if something bizarre took place, and I'll mention this does happen, where some recombination took place between the two elements, then you might see that as well by perhaps having both markers. And you can look at this both in males and females, of course. And in females, it all sort of makes sense in that 50% of the alleles are the donor chromosome. The others add up, obviously, to 50% too, roughly. And among those, the predominant classes are the eraser copies, as you hoped it would. It leaves the gene drive in place, which you wish it didn't. And then there's this percentage here where it didn't copy. And an important feature to notice here is that it didn't copy in females, that there's this big group of that are delete but not replace in females, but far fewer in males. And that suggests, because these genes are on the X chromosome, that those that are deleted have damaged the chromosome very, very frequently in females, like 10 to 1. And there's just a very small minority of cases in which the deletion involves a much more local event, which can be then viable in males. And that's reflected in the overall transmission, in that in males, you have a much higher percentage transmission of the eraser, because there's this whole category of individuals in females that are missing in males. And so these do not have the eraser. And so you have a higher percentage of males that carry it because they are missing this group. So that's already an indication that there's that damage that results in male lethality in this category, but that can be sustained in females because they've got one good copy. So I'd like to just kind of briefly go through this paper, which again examines this with multiple different erasers and eCHACRs that I'll mention soon as well, in all different kinds of configurations. But I'm just going to summarize the key points here. But I do encourage those of you with an interest in this to look at that paper, because we've devoted an enormous amount of effort to doing this in every possible way we could to test all these different possible permutations of this kind of experiment. So one of the things you can see right away is that if you look at males versus females, as I mentioned, that there's a higher transmission frequency in males, which again, I was explaining potentially by the fact that females can carry a damaged chromosome, but males cannot. And so that was the basis for it. Now, one of the questions would be then is that if you look at the donor chromosomes versus the receiver chromosomes, which do you see the bias in? And if you're looking at the receiver chromosomes, you're looking at, well, how often does it copy? And it turns out that it copies equally well to males and females, which makes sense, really. There's no real reason why it shouldn't. And that the difference is in how often are you transmitting the donor chromosome. And that, again, is kind of consistent with what I was pointing out. And another point that goes with that marking the donor versus receiver chromosomes is an essential point. And here, again, what you see is that if you look in males, in females, you get equal inheritance of the donor versus receivers, because that kind of makes sense, the donors being in gray and the receivers in orange. But in males, you get a much higher transmission of the donor chromosome, because you're croaking or compromising the viability of those chromosomes, those target chromosomes in males. It's a high degree of male lethality. That's the hypothesis that's supported by this data. If you look at how often do you leave the gene drive in place, again, that's not too surprising that it's the same in males and females. And that's about 10 to 12% of the time. It turns out, and I don't have time to go into this, that there's a significant fraction of these, maybe a third of these, that also have some damage to them. And you can see that a little bit here by the fact that there are slightly fewer males than females. And so the take-home message here, lesson 2, is that erasers aren't perfect. That's for sure. And then I mentioned, well, let's just focus on that group that is in females, where you get deletion but not replacement. As I mentioned, there are many more of those in females than there are in males. There are fewer of those in males by a factor of about 10. And the hypothesis is that in females, you can tolerate some chromosome damage, like a deletion, for example. The Y locus is right at the very tip of the X chromosome. So you can imagine perhaps a deletion of the tip of the X, or some fraction of that region that contains, say, an essential locus, so that in females you can live because you got the other good chromosome, but in males you can't. And so what we did is we took a set of duplications. We isolated females that carried these, that were like this, that carried such alleles. We isolated those, set up lines, about 10 or so of those lines, and then crossed them to different duplications of the X chromosome. And it turned out that a duplication that just covered the tip of the X chromosome rescued the lethality of all these, so that we could then acquire males that had these lesions in them. So it's, again, consistent with this idea that there's localized chromosome damage to the tip of the X that is sufficient to make the chromosome not viable in males. So erasers can damage the target chromosome, and they do so at a fairly high rate. In fact, if you go back to look at the numbers, it's about 45% of them in this particular case, on average, between 40 and 45%. Now, the other thing is that we wondered, what is going on with this damage? Is it because you're cutting the chromosome twice because you have 2 guide RNAs, or is it something in the process that is different? That's some other aspect of it. So, for example, one thing that's different from an eraser than a gene drive is that you only have homology on one side of the element at its point of insertion for any one side that you look at, whereas with the gene drive, the gene drive is inserted neatly into the genome at a single spot. Whereas with an eraser, you delete the region that would otherwise carry the gene drive. So you're cutting the gene drive out. So they're different in that respect as well. And so what we did is we made these. This is a cartoon then of the situation where you have the 2 guide RNAs that work to cut the gene drive out on both sides. And then we made single cut erasers where we injected both guide RNAs to get them inserted into the genome, because you have to do that to get them in, but they only carried genetically one of the 2 guide RNAs. So this one only carried gRNA2, and this one only carried gRNA3. So this one only cuts on this side, this one only cuts on that side. And so then you look at the prevalence of phenotypes. And then if we start off, this one's easier to analyze. Remember, the big thing with the eraser is that you had this big population in females that were double negative. That is, they didn't have the gene drive marker and they didn't have the eraser marker. And those were the ones that had these damaged, deleted areas. And in fact, with guide RNA3, you see that is exactly what takes place. You have this big population of females that are double negative, but very few males. So that was consistent with that. And then if you look with the other one, it's a little bit more complicated. You don't see it as immediately, but what goes on here is that if you look at the GFP positive group, there are fewer males. There's a smaller percentage of males that are GFP positive only than females. And what's going on here, we think, is that if you cut here, the damage is happening distal to the cut site. So that leaves the GFP in place. Here, when you have the cut site, it takes the GFP out. So this is the category that is a situation where you're cutting over here, taking out the GFP. When you cut over here, you leave the GFP in. So what that means actually is that it's very precise, at least on this end, where the cut site, where the damage is. It's always distal to the cut site. And in this case, it leaves the GFP in place. But again, you see the same kind of decrement. So the conclusion then is that it isn't just that you're cutting with two guide RNAs that's causing the problem. It's that there's a mismatch in homology. That is, you have the homology coming up to the cut site on one side, and then you just have the element of the eraser. And then on the other side, you've got the gene drive, but you don't have homology on the other side. Whereas with the gene drive, you have homology on either side of the gene drive when it cuts the target chromosome. So that's what we think. We've not seen very many examples of where we think single cut gene drives are causing damage. There are a few though, so that's something you always have to bear in mind. And it's something I think that is a very important lesson, again, that we've learned from our studies, is you must mark the donor versus receiver chromosomes so you can parse these kinds of things out. A very standard thing that we did ourselves at the beginning was just to make an estimate of what the conversion frequencies were based on the assumption that you get Mendelian inheritance of the donor and receiver. But as you can see, that is an assumption that is, in this case, not at all close to reality. And so you have to take this additional step to mark the donor and receiver chromosomes to be able to come up with a more accurate description of what's going on. In addition, I'm not going to go into this in much detail. We did spend quite a bit of time in figuring them out, though. So if you look at the paper, you can satisfy your curiosity if you're interested in it. But there were some weirdnesses where we would get the product of the confrontation would be progeny that had both the red and the green marker. That was consistent with some type of recombination event taking place. And in fact, in males, the category that were double negative, they were also a recombinant class that had to do with recombinations that were just outside of the element with sequences that were inside of it. But these make up only 1% to 2% of the population. And in each case, what they involve is some type of event where a sequence in one area undergoes illegitimate recombination or partial homology-based recombination. So there's homology, but it's not the homology at the site that we want it to be. It's, for example, here it's the guide RNAs can recombine. So that's the target site for the guide RNA can actually recombine with the region that is directive from the guide RNA, that 20 nucleotide piece. And that 17 base pairs that's on the one side of the cut site, it can recombine with the equivalent sequence that's carried inside. And there are all these variations on that. And some of them, in fact, don't even require homology at all. So that's a very low percentage, but there's some percentage of them that just are based on something we can't quite figure out. We've sequenced them, but we don't know exactly why. And so that sort of take-home message here is that these illegitimate recombination events that do proceed in the same way that copying does by the D-loop SDSA branch of homology-directed repair. So the repair mechanism is the same. It's just that you get sort of template skipping, where a piece of where there's a region of homology that pairs up with a small region of homology someplace else, and it can be on the order of 10 bases or less, even that can mediate that. Now those are pretty rare, and you can do the best you can to make it difficult. You can flip sequences around so that if that happens, and you destroy the chromosome so you don't get it out the other end. So there are various strategies, and that was done in ERACR-2. We didn't make all those manipulations to try to reduce that. And you can reduce it to like 1% or so, but you can't, I think, at the end, eliminate that category at all. So that's something to bear in mind. Now, we did test these, though, to see how well they worked, all these different variants, and none of them performed very well. Some of them didn't do anything at all, and there were a few that could copy that had both elements, but they did it in a very inefficient way. And there wasn't anything that we saw from that analysis that gave us reason for concern. Now, of course, we didn't see everything, and so you just have to bear it in mind. And I think it also speaks to a point that I've often raised in terms of how you might think about a gene drive registry and so forth, where you want to make sure that all the genetic elements that are out there are documented so that it's like the space junk, which is now dominating the thoughts out and sending satellites into space. There's so much trash out there. You keep worrying that one of those pieces of trash is going to destroy a billion-dollar satellite up there or something. And so this is the same kind of worry, I think, where you want to make sure that if you put multiple genetic elements into an organism, that they're not conflicting with one another, and you don't get these kinds of unwanted forms of recombination between elements that you've introduced without thinking that they might eventually interact. They might. And so I think you want to be careful about that, and that's one of the key elements that I would include in a sort of gene drive registry issue, is just to make sure that you document all the little pieces that are in there so that everybody knows what's out there, and do everything you can, if you're the N+1 version, to minimize any homology to those that already exist. So how do they work in cage trials? Well, we did it in two ways. Remember, we had an eraser that was on equal footing with the gene drive. It was yellow minus like the gene drive, and they both suffer this problem that yellow minus males are at a competitive disadvantage with regard to mating with yellow plus females. And then we also have the ERACR-2 version, which has the recoded version and therefore restores yellow function, and therefore should benefit from that ability to mate with yellow plus females. And so this is an example of 3 cage trials, or 4 cage trials, I guess here, that went 10 generations with the eraser in red and the gene drive in green, where it's the yellow minus against yellow minus. And that just looks at how good the gene drive is. And I have to admit, I was a little surprised, given that there are all these various things that are going on, that it drove as well as it did. Now, of course, the damage class is predominantly male lethal, and so it's not too surprising that that group drops out. And so they kind of go together with the gene drive copying and getting rid of the gene drive. But it did perform better than I would have expected, just based on those numbers from individual pair crosses. And we think that part of what that is that there's a bit of a fitness cost that's also associated with the gene drive that is potentially leading to this somewhat better than expected performance. But now when you put the eraser together with that has the advantage, the fitness advantage of being yellow plus, now it runs like a little Maserati. It goes right to the 100% completion within 6 generations, totally getting rid of the gene drive. The other thing too, if you recall from the experiments I was talking about with the allelic drive with DDT, if you look at the variation here, and the modeling variation reflects that as well, based on all the parameters that come from the testing. This is done by John Marshall's group. Jarrett Bennett and John Marshall's group spent an enormous amount of time making these charts that have like 150 genotype type entries into it. It's amazing. For each one, he gives a probability for what it's going to do from the data coming from the single crosses, and it reflects this standard intercage variation. This is typical from when you do studies. There's this intercage variation, and it's predicted then from the modeling as well. But when you look at it, when you have these 2 selections, it is much tighter. And the modeling suggests that it should be 2, because again, what you're looking at is the probability of joint failure of 2 independent processes, the drive process on the one hand and the selection for yellow on the other hand. And because they're totally independent mechanisms, you get this sort of cooperative reduction in variance. And so that's an important thing. That means that the performance is more reliable. So erasers can drive very efficiently, particularly when recoded, where you have this dual drive capacity. So chasers, again, another backronym, is an idea where an element can chase after a gene drive because it has a guide RNA to do that, but no Cas9. It's a split drive sort of situation. And the idea is that if you have a gene drive in one place, you make what we call an eCHACR that can copy itself in another location through a guide RNA that cuts at its site of insertion, but it also carries one or more guide RNAs that work to mutate the Cas9 at a catalytically sensitive region so that it's hard for the Cas9 to escape its action. And in fact, we did test 2 different guide RNAs, not at the same time, but individually. And each of those guide RNAs, when we analyzed them, eliminated Cas9 activity essentially 100% of the time. In fact, it was so efficient that for one experiment, we actually wanted to have a Cas9 resistant form of the mutant form so that we could do an experiment it's actually in the paper. And we couldn't generate one experimentally. So we had to go in and design it on purpose and inject it as a recoded version of Cas9 that couldn't be cleaved because we couldn't generate one with an eCHACR. That's how efficient it is. So anyway, eCHACRs in the white locus, we put them in different locations. You can, again, in the paper, you can look at that. But the one that did the best was it inserted in the white locus, which is also on the X chromosome. And it copied over 95% of the time. And it inactivated Cas9 by various phenotypic measures over 99% of the time. In fact, when we sequenced all of the alleles that came out, 100% of them were mutant. And again, as I mentioned, when we tried to extract a mutant allele that had been acted on, we couldn't get one. So it probably is actually closer to 100%. But anyway, they can work. The good idea is that for an eraser is that in principle, it should work on any gene drive anywhere because it's the erasers the eCHACRs at a second location, whereas the eraser has to be built at the location of a gene drive. So if you want to get rid of a gene drive for some reason, you have to know where it is to use an eraser because you have to put the eraser at the same place. Whereas if there were some reason to get rid or halt the spread of a gene drive, an eCHACR in principle should be able to do it no matter where it's inserted, as long as it's targeting the same Cas9 form. And of course, if you look at the benefits of it, one of the benefits from the eraser that's obvious is that it's easier to do the recoding using the endogenous locus, where you just have the endogenous expression of the endogenous locus producing an endogenous protein in its normal pattern. You could, in principle, put a cDNA, a rescuing cDNA, on the eCHACR, and there have been quite a few publications on folks that have done that kind of sort of cleave and damage the target and have a recoded version that rescues someplace else. But then you're not providing the gene in its normal pattern and from its normal location. And so it's an advantage of the eraser in that case to be able to provide it in the normal pattern. So this is an example of an eCHACR in white, the one that performed the best, across from a gene drive in yellow, and just kind of indicating with these arrows all the things that can happen. The simple thing, of course, is it can copy itself into the white locus, the eCHACR. Then it has a guide RNA that mutates the Cas9. Then the gene drive has a guide RNA that can cut the yellow locus to copy itself. So if you look over here, you can see, in fact, I was worried that wouldn't work at all, because you can think, well, now you've cut all 4 strands of the DNA, right? One thinks that the copying mechanism happens at a 4-stranded DNA stage after replication. And you could worry that all of the strands got cut, these cut here and those cut here, and so that there might be no viable product to repair from. That was a concern that I had. And that's surprising that doesn't happen. However, as we'll see, this cut is a more difficult cut to repair than this one. And the reason is that this one, as again, as in the case of the eraser kind of story, is that the gene drive cut has homology on both sides of the cut, whereas this cut doesn't have any homology on the other chromosome. If you look at this region over here, the Cas9 and the sequence, there is no corresponding sequence on this side. So this is harder to repair than this, as it turns out. So if you look at the overall performance, and again, we've done this with static eraser, with static forms of Cas9, as well as the driving forms with eCHACRs on different chromosomes. So this is just one of many different examples. And so I'm summarizing to some degree the various results. But one of the key effects of certainly of this one is it copies very well. This is the best copy when it copies in some cases like 98%, 99% of the time. So this is a very efficient copying one. And the other thing is that it's very, very good at eliminating the activity of Cas9, because one of the ways you can estimate the activity of Cas9 in terms of the recovered chromosomes is if this element copies, it makes a yellow minus allele. And so if you cross that in this crossing scheme, you will get yellow minus every time this thing copies, or if it's active at all, even to create a mutation on this chromosome. So if Cas9 is active, this chromosome will be mutated. But what you see over here is that in males, where you would be picking this up, there is 0 yellow minus phenotype coming from this activity. So the Cas9 is, in this experiment, 100% activity is eliminated. So that means that this guide RNA is working very, very efficiently. And as I mentioned to you, it was so efficient that we couldn't even identify a Cas9 mutant form to recover from this type of experiment. And we looked through thousands and thousands of progeny. Okay, so the lesson here is eCHACRs copy very efficiently and eliminate Cas9 activity approximately 100% of the time. The other question is, well, what about its effect on the drive? And so the drive by itself in males and females copies about 70% of the time, and with a transmission of about 85% transmission, whereas the allele, this version over here in this configuration only copies itself about 10 to 12% of the time. So it's about a 5-fold reduction in the efficiency of this being able to copy. And we recovered, as I mentioned also, we recovered a significant category of these elements that were cleaved, that were GFP minus, suggesting that this whole area had been taken out, and that were lethal, which could then be rescued by crossing to that duplication of the tip of the X, suggesting again that there's difficulty copying here because this is hard to repair, and the repair failure often results in a local chromosome damage that is part of this process. So that's another feature of this system. So eCHACRs reduce copying of the gene drive, which is a benefit that's kind of thrown in. We didn't, when we designed it, we didn't think of that being necessarily an effect, but it's a positive effect for their behavior. Then again, as I mentioned, there's this issue of can they reduce the percentage of these GFP guys, and you get this category, as I was mentioning, this fairly significant category, 15% that are EGFP minus. And again, you get this bias in the inheritance of the donor versus receiver chromosome. So you're inheriting fewer of these white apricot chromosomes here in orange than the target, than the opposite chromosome. Again, consistent with this idea of chromosome damage. And again, you can take these animals here that don't have, these individuals here, you can isolate these mutations that have resulted from this action, and they're lethal in males, but can be rescued by the tip of the X. So that's another thing. So eCHACRs can also damage the gene drive chromosome. Now, something that's a little bit more subtle as an effect is that if you're going after a situation where the Cas9 source is not on a gene drive, so it can't copy itself, so you don't have that problem with 4 strands being cut and so forth. So you're just looking purely at the action of the eCHACR in this case, just distilling that aspect of it. Then you get very good drive. But again, you see this bias in males of inheritance of this donor chromosome here, relative to those that it's copied on, which will have dual fluorescence because you have both red and green if it copies. So if this is basically the donor chromosome, those that are just GFP only. Here, though, there was no lethality. That is, we could collect the females over here that in principle would have this problem, whatever it would be, and then ask, well, do we see again a fraction that are inviable in males? And we never did. And so this is different from that other kind of damage where, a deletion or something got generated on the target chromosome. This is something that happens just during the drive process itself. And we're not entirely sure what this process is, but this is a fairly significant effect. So eCHACRs that cut a target chromosome twice, and that's the other thing, is that we put them on different chromosomes. If you put it on a different chromosome, it doesn't do that. So it has something to do with cutting the target chromosome twice, but it's something that once it gets through, whatever it is not heritable as a characteristic. So it may be a transient form of DNA damage that gets repaired, but for whatever reason, lowers the frequency with which male gametes get through in that first cross. So that's something that we need to do more work on understanding why. So there again, we did the cage studies on this, where we crossed the eCHACR to the gene drive, and what we found is that the eCHACR drove to completion by 10 generations, where 100% of the Cas9 activity, shown here in yellow, one of the features of the activity of Cas9 is it causes mosaic eyes if there's a wild type allele in the background. And so that ability to do that peaked as they met and mated, but then disappeared. And that by the end of it, 100% of the sequenced flies had mutated Cas9s, and they were homozygous for the eCHACR element. So eCHACRs drive very efficiently in population cages and eliminate Cas9 activity. And something we don't understand is we carried that out a little bit further than we did for the publication of the paper, and we can't quite understand why it is that the gene drive, which goes up initially, that makes sense, it would copy itself, then it starts coming down. Well, it could come down because, as I mentioned, the eCHACR can cause some damage to it, so that could be part of what causes it to come down. You kind of at first, my prediction had been that what would happen is that the gene drive would kind of go up to some point and then stay flat because the eCHACR isn't trying to cut it out like the ERACR is. But again, because it's creating that damage, I could understand that there'd be a dip like we saw down here. So this level is kind of consistent with the modeling. You can see there's a lot of spray in this, but it's kind of in the middle of where the modeling would be. But the modeling does not take it down, based on the parameters that we use, does not take it down to zero. So we don't understand what is going on there. And it could be that there is, we know, a fitness cost for the Cas9. However, we've done that kind of experiment separately. So we would have expected if from this point on, just based on the fitness cost of the Cas9, it should kind of go down like this, not like this. So we don't quite understand that. Why does the gene drive disappear, and why does it disappear at the rate that it does? But in any case, these are things that are all working in favor of the system. And so even though we don't quite understand why they're doing what they do, they are working in the direction we would like them to. So what are the lessons then, in summary, that we've taken from all this? Marking the donor versus receiver chromosomes is really important, because in the case of the ERACR, it's causing about 40% damage to the target chromosome. You can't ignore that. That's a significant percentage. ERACRs are not perfect. It leaves the gene drive in place 12, 15% of the time. A significant fraction of that, though, a third of them, we think, actually are damaged, and so they will go away too, but still, it does leave some in place. The ERACRs can damage the target chromosome. That's what happens in that group where they don't copy, where they delete the ERACR, they delete the gene drive and don't copy. A very significant fraction, 90% females, are these damaged chromosomes. That the two guide RNAs are causing, it's that the reason that the damage is being created isn't that you've got two guide RNAs that are cutting and creating a difficulty in joining because they're cutting at the same time. In fact, we think they're probably cutting in a staggered fashion, one after another, each one creating a problem where there's one-sided homology, and that has a certain probability of leading to a failed repair event that leads to something that's bigger. And we haven't analyzed exactly what that is. It'd be interesting to know, but it's localized in the sense that it can be rescued by a short local duplication of the tip of the X chromosome. So it's not destroying the entire chromosome. They also, erasers can generate unintended products through sort of SDSA D-loop forms of illegitimate recombination, or it's real recombination, but it isn't happening between the sequences you want it to. So if we've got these even short sequences of homology, and that has to do probably with template switching, which people have shown is a very frequent companion of HDR, homology-directed repair, when using exogenous templates as well. So the polymerase skips between the two templates, and that can create these partial pairing or products. Erasers drive very efficiently in population cages, despite their imperfect performance on these individual measures, particularly if you give them a second edge. And in general, with the data from the allelic drives with respect to insect, the KDR mutation there, again, we saw a combined benefit from having two different drive mechanisms. And they, eCHACRs copy quite efficiently and eliminate Cas9 activity 100% of the time. They also can significantly reduce the copying of the gene drive, in part by damaging that chromosome, and which is that point there. And they can work very efficiently in population cages, completely eliminating the Cas9 activity, and then somehow, at least in the case that we looked at, they went even further and got rid of the drive altogether. So the questions that remain is, what is the nature of the eraser-induced damage to the chromosome? We know it's local. Does it take out one or two genes that are essential? Does it almost take out the whole tip? We've tried to do a little bit of cytology to see what goes on there. It doesn't look like it's taking out the entire tip, but more work would need to be done to delineate that. But that would be certainly interesting. We have this sense from the single cut data that the cut on the lesion on the centromere proximal side happens at the cut site. So it seems like it goes from that location to someplace toward the tip. So we can potentially create primers that would allow us to see that based on that kind of realization, but we don't know yet what those forms of damage are. What is the nature of eCHACR-induced damage to the gene drive chromosome? We don't know. We know that it can take out the closely linked GFP. In that case, that's centromere proximal. So it may be a kind of different mechanism there. It doesn't look like it's just going to the chromosome tip, because if we're just going to the chromosome tip, they'd all be GFP positive. So the fact that there's a significant GFP negative group suggests that in that case where you're cutting all four strands, the damage is somewhat different. It's a different genetic situation. It's a different repair problem. There are different outcomes, we think, that come from that. We don't know how eCHACRs that cut the target chromosome twice bias donor inheritance. It's not due to damage, or long-lasting damage. It's not due to heritable damage. So we don't know what the basis of that is. That's interesting, but it's significant. It's 2-to-1, so it's not trivial. And we don't know why the guide RNAs. I didn't mention this, but in the case of the single cut erasers, we asked how well do they work individually, compared the guide RNAs to them together, and the answer was, if you're using them to cut out a big gene drive, they don't work individually as well as they do together, and there's some kind of cooperative action of them together. One works a little better than the other one, but neither of them work as well as them put together. If the distance between them is shorter, so you can use an eraser just against a wild-type chromosome, because a wild-type chromosome has the cut sites for the eraser in it too. So if you have a form of Cas9 someplace else, you can ask how well does the eraser copy in this what we call kind of copycat mode. And it it copies quite well. And there you can look at how well do the single cuts work. And they work much better than they do when they're trying to cut out the whole bigger element. So that cooperativity between the guide RNAs seems to be size dependent. It's much more cooperative if they are working on a big element. And maybe it's because when one cuts, the other's more likely to cut. We don't think they cut exactly at the same time, but maybe there's something about the repair process that if you stimulate the repair process on one side, then it's easier to recruit the repair machinery on the other side. We don't know, but that's another interesting question. But it's another area for future work. Future goals are to test erasers and eCHACRs, of course, at the different release ratios. That's an interesting kind of experiment to do. We just, imagine that the eraser, the gene drive had gone to completion, and then to challenge them, what would happen if you did it at sort of intermediate stages. Of course, to build these elements for mosquitoes where they're intended, and to develop two-dimensional models. Some of the modeling that has come out of the Imperial College group on 2D modeling, Ace North's beautiful analysis with his colleagues. It'd be nice to model that as well in terms of these elements, and then maybe to create certain kind of experimental systems that allow that kind of similar 2D modeling to be tested in an experimental situation. And then, of course, responding as part of this whole presentation is to make sure that folks in the community know the facts as we know them, as best we can tell. And are there questions? Are there concerns? Are there additional experiments that we might think of doing based on feedback we get from the community, which of course is the heart of this whole process. So I'd like to end just by, again, acknowledging Valentino Gantz, the person in my lab who started all this. He made the first gene drive in flies and then in mosquitoes, and played a huge role in everything else, including starting the original ERACR, eCHACR experiments himself. And he has had a wonderful career since he left my lab, starting his own operation where he's gotten grants and papers in top journals and so forth. He's a wonderful human being and a phenomenal scientist. Adriana Adolfi, who is a collaborator from Tony James's lab, working with Gerard Terradas, did the recoded drive, a large work of the recoded drive. Annabelle Guichard is the person who initiated the work on allelic drives, showing them in principle. Bob Grashri is the person who led, together with Raja Kushwaha, the experiments on the KDR story. And again, the ERACR story, which is was led by Shannon and Emily, combined with these other folks that put in an enormous amount of work. And again, I encourage you to look at the full paper that has all of the in-depth analysis of all of the different possible ways of doing experiments. And of course, our collaborators, in particular, Tony James at UCI, Adriana, again, being a member of his lab. And, we've been working together from the very beginning on all this, and John Marshall doing the modeling. He's been a great collaborator there. And then, of course, I acknowledge with gratitude the support we've had from the National Institutes of Health, the Paul G. Allen Frontiers Group, and a gift from the Tata Trusts, as well as money from the DARPA Safe Gene Drives Initiative. Thank you.
Thank you. Thanks a lot, Ethan. A real tour de force, and really fascinating for sure. We have some questions, and we'll get to them in just a second. I just have a couple of my own that I'm going to privilege myself in asking first. So, I mean, there were a really lot of interesting observations that you made along the way from the genetics. And I guess one of the things that, and tell me if my interpretation is wrong here, but despite all of the genetic phenomena that were taking place that you were able to dissect because of all the markers that you used. When you actually put these things in cages, they, at least from both systems, both the ERACR and eCHACR systems seem to work, right? Is that, I mean, and in some cases very, very well. Would that be a fair assessment?
Yeah, to be honest, David, I was a bit surprised at how much better they worked in cages. I was feeling like, well, will they mate properly? I figured the cages would be a harsher test than the pair mating crosses, and yet they seem to perform better than I would have expected. But to be fair though, with the modeling, when the Marshall group took the parameters from the pair mating, the single generation crosses, and then did the predictions, those predictions matched very well with the cage studies. So those performance parameters, with some exceptions, like, we don't quite know, for example, in the case of the eCHACR, why does it completely go away? The modeling didn't explain that, but it was consistent with their good performance in the cages. So to some extent, it may be somewhat non-intuitive why they work as well as they do, but the modeling in the end was predicting that it should be like that.
I guess the other thing that when you were describing these systems, the multiple cutting that was going on in these germlines that you were working with, I guess, sort of made me think that I would anticipate that there had to be some recombinations and all kinds of other things going on. So, and I guess a specific question had to do with your allelic drives as to whether or not linked versus unlinked, if that would make any difference. It just seemed to me in a case of a linked, in linked situations, you might actually be having a higher percentage of generating maybe deletions. But anyway, so I just, what do you think of that?
Yeah, I don't know, David. I mean, I don't think so very much. We did notice in the case of Annabelle Guichard's original allelic drive paper with Notch, that it actually increased a recombination with an intervening marker, that is Notch was in between it and another marker, and it increased the recombination between white and that marker twofold. And this is kind of, people have noticed that in yeast as well, that cutting the chromosome results in a modest increase in recombination. But presumably that's just because when you cut it, there's an opportunity to undergo a recombination event that it's pretty low. But if you look at the target sites that we've been looking at, like at yellow and so forth, there may be an indication here or there that there's a small percentage increase, but it's really not much. It's at most a 50% increase in the recombination between these tightly linked markers. So we don't think there's much recombination going on, or induced recombination that's going on from the cutting. And in the case of the KDR drive, they're on the very opposite ends of the X chromosomes, so they recombine freely. So they segregate independently. And again, that behavior was similar to the behavior of Notch, which was much closer. And so I don't know. It's a good question, David. I mean, I don't want to be, I don't want to make a sweeping statement based on just a few observations, but we don't really have much indication that there's a huge effect on recombination. Say it increases it twofold. I would say that's the maximum rough estimate, and maybe less in many cases.
Let me get to a couple of questions here. Let's see, Hector Kamada asked, what are the likely remnants after an eraser deletes the gene drive?
Well, if it deletes and doesn't copy, well, I think based on the single cut analysis, one side of it is going to be at the cut site, because, if you look, for example, at the one where you're cutting on the centromere proximal side, you get the same number, the same percentage of females that carry that are basically double negative, that don't have either fluorescence. That is, that basically haven't had, that just have a deletion, that just have the gene drive's been cut out, but that's it. There hasn't been a copying of the element. And so that would suggest then that and then if you go to the other side where you use the one that's cutting on the telomere side, you have almost an equivalent percentage that are GFP minus. Now, a fraction of those might be GFP negative too, and it goes a slight distance. So you can't say for sure that they're 100% at that border, but a very significant fraction of them seem to have a cut site that's defined by where the guide RNA is cutting. So if that's true, the hypothesis would be is that you cut there and then there's some kind of large resection process that goes on, or some type of, my guess would be is that it's some kind of weak homology based thing that we see with the erasers too, where you get a little piece of homology that may be some distance away. And so I don't know, is it a target sequence that's a little bit like something that's near yellow that keeps recombining with it? And so there's an intervening essential gene in there and it takes that out and it's always the same kind of borders? Or is it that second border can vary from one individual to the next? And until we, get those sequenced, which we could. I mean, you could also just do deep sequencing to try to find out what the borders of those are. So those are good questions for the future. So I don't know, but I suspect that at least one side of it is anchored.
Yeah. Okay, I'm going to move on to a question here from Pratima Chanuri at Texas A&M. So with erasers, there is a serious skew in a number of male progeny. Do you think this would affect the transmission rates of eraser elements, given males are generally a better way of transmitting transgenes?
Well, in general, you're better off if you use a gene drive and any drive system on an autosome, because of course you can get active copying on both in males and in females. But in a situation like this, where a lot of the mistakes that are being made are mistakes that are lethal to the chromosome in males because of it being on a sex chromosome, I think you do pretty well with the eraser. Would a comparable eraser setup on an autosomal gene drive target work better? I would think so, but then the gene drive would work better too. So what would the balance be? And in fact, that's a really interesting thing to do in the future is, for example, I didn't have time to talk about it, but Gerard Terradis, who was working with Adriana, has also published a nice study where he looked at a bunch of recoded split drives that are using the same principle as in Adriana's paper and in the study and in the recoded eraser. And he found, for example, that the pros alpha locus, if you recode that, he gets almost 100% transmission through both males and females in that situation. And we have now another one in another essential gene where it's about 100%, very close to 100%, with almost no mutations getting generated. So it's a little bit like Tony's. And so those would be good examples of places to make erasers and eCHACRs against those, because they would be much more challenging drives. And again, they're in an autosomal context. And so it'd be interesting to see how the balance would play out in that situation. But yeah, that's a good point.
Yeah. I'm going to. Yusuk Lee has a question here, and she says, "Great work, Ethan." And as for the regulatory approval for field deployment of gene drive organisms, do you think one has to come up with a working eraser as a mitigation strategy before a field trial of a proposed gene drive system? So this is a general question.
Right. And I'm really glad she asked that. Thank you. That's a really,
some sort of a mitigation.
Really glad you asked that, because when I had my 15 minutes in front of that original NAS panel that did a great job, I think, in looking at the future of gene drives and so forth, I made the case that you should not have to do that. And Harmit Malik, I think, made a similar comment at some point in one of his commentaries, which is that if you have a situation where you don't trust the gene drive, and then you add an eraser or some other mitigation system, that you also could have some unintended consequences. Now you've got two things that have potentially unintended consequences. is that a reassuring thing? And the answer is no, I don't think so. And so my view is that if you're interested in in a regulatory process, if the application is to say, we believe this gene drive is safe enough to use to test it out, then that should be the criterion by which you judge whether or not to go forward in a regulatory basis. Does it make sense to build these mitigating elements on the side? Sure, but it shouldn't be part of the regulatory approval process, because if your argument is, well, we're going to put this kind of iffy gene drive out there, but good news is that we've got a system to remove it. I think that's a very uncomforting approach. I think it's a much better thing to say, look, we believe in the gene drive as it is. We think it can. We think we can confine it like on an island or whatever the application process is, and with the right approvals and stakeholder engagements and all those very important components. Then I think that's fine. Does it make sense to have it be part of a target product profile where you have something that can take it out? I don't think it should have to be part of the TPP of the gene drive. I think it might make sense to build one and have a TPP, or at least a description of what a mitigated system might be. But I think no, I'd be strongly opposed to having mitigation systems of these kind be part of the approval requirement, because I think the inherent logic of that is flawed, which is that if you feel that the gene drive is providing some kind of risk factor that you're not comfortable with, then I don't see how providing one of these imperfectly acting mitigation systems is going to alleviate that. On the other hand, there are other kinds of scenarios that should be considered, in which these kind of mitigating elements, one might be happy to have them on hand, particularly eCHACR type things that are more general. But an eraser can be built pretty quickly too if you figure out what the potentially problematic drive would be. So my answer is no, I would not make this part of the TPP or the required approval process for the gene drive itself. But I think it certainly wouldn't hurt in terms of, how it looks to the community, how serious you are about safety and consideration that these experiments, for example, have been done. That's one of the reasons we spent so much effort on these experiments, is to make it clear that we take this kind of thing very seriously, this issue that David brought up at the very beginning very seriously. And so that's why these were tested. And of course, as funding and time and everything permit, we will do everything we can to build these kinds of elements in mosquitoes also. But I would not predicate the use of a gene drive or a test of a gene drive in an open field test based on these. No.
Mm-hmm. Good, thanks. Pratima Trinuri asked sort of not the same question, but a related one that has to do with perceptions and ideas that regulators and the publics might have. And first of all, she says, amazing work, thank you. And a rather broad question here, your thoughts on how the general public and regulatory agencies might react not only to transgenic gene drives, but to what seems to be some cases of rather untraceable recombination and damage events carrying different combinations of transgenes. So I guess it's more of a question about about the genetic phenomena that you were witnessing
right.
During the analysis and the effects that might have on
right.
people's perceptions.
No, those are good questions, and that's one of the reasons why we went through the trouble of recovering some of these damaged products from the confrontation of the eraser with the gene drive, and then testing those subsequently in crosses to see how they performed, to see if there was anything disturbing or whatever. And all that they were is not as good. And if you have two different markers instead of one, and it's bigger and it's less wieldy and only part of it copies, those kinds of things were what was going on. They're just not very good drives is mostly what they are. But yeah, you have to worry about that. But if you kind of step back and you look at the big picture, let's compare these gene drive elements and the erasers to say something like a P element or a transposon, and what those do, like say in PM dysgenesis, where you have 20 of them that go out, wiping out the genome of the recipient element, causing massive amounts of chromosome, damage as they're doing so. And you compare that sort of genome violence, vandalism, to this relatively precise, localized problem. Every case we looked at where there was chromosome damage, it was localized to the area of the tip of the X chromosome. It didn't go everywhere else in the genome. We could rescue it always. So it wasn't like we damaged something on a different chromosome or a different part of the X chromosome. So I think that it's important to have this sort of high resolution microscopic view of what exactly goes on with these elements, understand, because as David points out, this is a new technology and it has this autonomous capacity that you want to be very careful in analyzing what can go on. But as far as I'm concerned, the news is good overall in that we haven't seen anything that is of mechanistic concern to me. And if you compare it to naturally occurring phenomena that happen all the time in various drive phenomena, like transposable elements being a very good example, this is orders of magnitude less chromosome vandalism than is happening in nature all the time. And, it's much more precise. It's much more trackable because you've got these fluorescent markers and so forth. And it's not to say that you can't get some unmarked things that wouldn't be seen right away. And it absolutely makes sense to go on paying attention to that. And for any given situation, as part of a TPP, for example, with a gene drive, I think it does make sense to ask, what are the off-target effects of that gene drive and how often and so forth. I mean, those are very legitimate questions to ask. But if you kind of look at it in the bigger picture of how does this compare to naturally occurring forms of genetic challenge, I think it's very modest. So I would not, if I were looking at this from a ecologist perspective or a broader view, I would not see this as reason for concern. I would be more reassured based on these studies than anything.
Yeah, good, thanks. I mean, this question of precision and off-target effects and these kind of things, I mean, these are really raging questions within the broader discussion of gene editing technologies, writ large, not just gene drives per se, but for every application of these gene editing technology, you can see these questions arising about these about unintended consequences or off-target effects or things like this. And I think this sort of is part of that discussion.
David, I noticed an interesting question that just came up there about. It says, in keeping with the P element example, do you think the genomes would start producing resistance mechanisms like piRNAs that do limit the spread of transposons?
Yeah.
And I think that's a really good question, and I wouldn't be surprised actually if that's true. And I think, of course, that's a kind of worry in the direction that I really do worry about, which is, will the gene drives actually work when you release them in the wild? Because that's something we don't know. We know that they work really well in cages, and the Crisanti group has recently done a remarkable study there in a big cage where they, found that their system, which is facing enormous potential pushback from the genome in terms of a single functional allele would give them trouble. They didn't find that, and they didn't find any other system that was limiting it. But on a bigger scale, if you start looking at a subcontinent or a continental scale, would you find things like mutations that increase the sensitivity of the piRNA system to take out the guide RNAs? I mean, people have anecdotally reported that if you put clusters of guide RNAs in a location, that they end up getting silenced. So that sounds like that kind of thing could be going on. And you could worry that as time goes on, that would happen. And that would be something particularly that you'd expect to pick up in a suppression drive system. In a modification drive system where you're not putting a lot of pressure on the mosquito, on the organism, I would expect it would be less likely to pop up as a problem, but you don't know. I mean, if the Cas9, for example, on its own carries a bit of a fitness cost, which in our experiments sometimes it does. Well, then maybe you would see that as a mechanism that crops up. So that's a good question, and it's one that, from the point of view of drive efficacy, I would worry about. But you will never know whether that is a problem until you're allowed to test it on a scale where it could reveal itself. And so that's why, I, along with many of my colleagues, have been, really hoping that we will be able to take these very good sort of systems that are now available and move them to the next step of a confined field test, so that we can obtain that data to see if, in fact, these kinds of issues come up. And if they do, are there ways around that? And that's a good question. So, for example, would it make sense to have some kind of component on the gene drive that, at least temporarily silenced the activity of some of the piRNA system? I mean, that might be a good strategy if that kind of problem were detected. It's a good question. Those are the kinds of things that really I do worry about.